This is the final video of your Test 5 material and the final video of the semester for you, so you made it. So we're going to talk about fluid electrolyte and acid-base balance, which is a great companion to the urinary system videos that you just covered. So the first thing we need to do is to talk about what osmolarity and osmolality is, because we're going to be referring to both terms in this PowerPoint. So osmolality, this is something that's maintained around 280 to 300 milliosmoles. And so what's the difference between osmolarity and osmolality? So osmolarity is the concentration of a solution, and you're describing it as the number of solute particles per liter. So this is a volume measurement. Osmolality is the total number of solutes per kilogram. So this is a weight measurement. So how many particles are in the volume versus how many particles are in the weight. And so that's the basic difference between osmolarity and osmolality. So osmolarity is calculated considering the volume of the solution, osmolality, the mass of the solution. The unit is moles per liter in osmolarity and osmoles per liter in osmolality. Osmolarity can depend on temperature and pressure, and osmolarity does not depend on temperature and pressure, which is why it's a little bit more of a practical measurement in this case. Okay, a rise in osmolality will stimulate thirst and cause the release of antidiuretic hormone. A decrease in osmolality will cause the inhibition of ADH. So let's consider the major sources of water intake and output for the average intake per day is about 2,500 mils. Your metabolism is going to generate about 250 mils of that. You'll get 750 mils or 30% through your foods, and about 60% or 1,500 mils through intake of beverages every day. Your average output, you'll lose about 4% of your water output per day on average through feces. On average through sweat, about 8% or 200 mils. You'll lose it from the skin, evaporation from the skin and lungs, about 28%, and about 60% through the urine. So let's take a look at the different ions and what their serum values are and what their consequences of these imbalances can be. So hypernatremia is an excess of 145 basically. Possible causes can be dehydration. It's very uncommon in healthy individuals. It can occur in infants and the elderly or someone who's unable to indicate thirst, but it can also result from excessive IV saline administration. So your consequences are basically going to be thirst. It can lead to confusion and lethargy, progressing to coma. You can have increased neuromuscular irritability. And so I went through a severe dehydration event last July when I did not hydrate sufficiently before walking in the Peachtree Road Race on a very hot day in downtown Atlanta. And so I just got completely dehydrated, and I can tell you that it definitely leads to confusion and lethargy. Hyponatremia is low blood sodium, and so you can get this through solute loss, through water retention, or both. You can get excessive sodium loss through vomiting, diarrhea, burned skin, gastric suction, excessive use of diuretics. Addison's disease can give this to you, renal disease, excess ADH release, and excess water ingestion. So this is what they call water poisoning that can cause basically hyponatremia. So you can get brain swelling here. If your sodium content is normal and water is excessive, you can get mental confusion, giddiness, coma. If this occurs slowly, muscular twitching, and so forth. I think as we go through these, I'm not going to read through all of the consequences and causes, just highlight them. Okay, so in excess of potassium is hyperkalemia. And hyperkalemia, basically, a cause of that can be renal failure or a deficit of aldosterone. So nausea, vomiting, diarrhea, bradycardia, cardiac arrhythmias, these can be symptoms of hyperkalemia. Hypokalemia Oh, and by the way, the reason why these get called kalemia is because potassium, its symbol is K. That comes from the Latin for kalium, the same way that natrium is a representative of sodium. Okay, so hypokalemia can be caused by GI tract disturbances, vomiting or diarrhea, and so forth, and consequences can be cardiac arrhythmias. So you can have consequences for cardiac arrhythmias if your potassium is too high or too low. With phosphate, hyperphosphatemia, a cause of that can be decreased urinary loss because of renal failure. Hypoparathyroidism can also cause that, and so forth. And your clinical symptoms can arise because of reciprocal changes in calcium levels. So basically, as phosphate levels change, calcium levels change. These don't directly cause problems. Hypophosphatemia can be caused by decreased intestinal absorption. Okay, chloride with hyperchloremia, possible cause of this is dehydration, increased retention or intake. Consequence, there aren't any real direct clinical symptoms, but this usually arises from sort of pH abnormality, which is going to cause problems on its own. Hypochloremia, a cause of this can be metabolic alkalosis because of maybe vomiting or excessive ingestion of alkaline substances. Sometimes an aldosterone deficiency, and same thing goes as with hyperchloremia. Hypercalcemia can result from hyperparathyroidism or excessive vitamin D, prolonged immobilization, renal disease, and with an excess of calcium you can have decreased neuromuscular excitability, which can lead to cardiac arrhythmias and arrest. And you see some of the other consequences here. Hypocalcemia, possible cause of that is burns or hypoparathyroidism, vitamin D deficiency. So you can get increased neuromuscular excitability, tingling fingers, tremors, skeletal muscle cramps, over the long haul, osteomalacia, and fractures. With a deficit in magnesium, or an excess of magnesium is hypermagnesemia. This occurs when magnesium is not excreted normally in renal failure, and you can get lethargy, impaired central nervous system functioning, coma, and so forth. Hypomagnesemia can be a result of alcoholism, chronic diarrhea, severe malnutrition, or diuretic therapy. And from this, you can get tremors, increased neuromuscular excitability, tetany, and convulsions. And tetany is kind of if you'll recall that tetany was a result of summation from nerve impulses coming in really quickly. If you have increased neuroexcitability, that can definitely lead straight onto tetany. Okay, so sodium is the most abundant cation in extracellular fluid, and they contribute 280 of the 300 total milliosmoles in extracellular fluid solute concentration. That means that sodium is your only cation that is going to exert a significant osmotic pressure. And it does so because it controls extracellular fluid volume and water distribution, basically because water follows salt. We saw that when we talked about osmosis in AMP1. So changes in sodium levels can affect plasma volume, blood pressure, extracellular fluid volumes, and intracellular fluid volumes. So sodium that leaks into cells gets pumped out against its electrochemical gradient, and so that's where the sodium-potassium pump comes in. And this means that sodium can move back and forth between the extracellular fluid and body secretions. An example of this is digestive secretions. Renal acid-base control mechanisms get coupled to sodium ion transport. So our homeostatic importance of ECF, or extracellular fluid sodium concentration, is that ECF osmolality will have an effect on blood volume and blood pressure. So the sensors that detect sodium concentration in extracellular fluid are osmoreceptors, and in the bloodstream it's baroreceptors. Your primary regulators of extracellular fluid sodium are going to be ADH and thirst mechanisms, And for the entire body overall, it's going to be the renin-angiotensin-aldosterone and atrial natriuretic peptide hormone mechanisms. So there aren't actually any known chemoreceptors that monitor specifically sodium levels in body fluids. And the reason why, again, is because the sodium-water balance gets linked to blood pressure and blood volume. So the mechanisms that control those, that control the changes in blood pressure or volume, these neural and hormonal controls actually do so by regulating sodium content. So in order to maintain homeostasis of blood pressure and blood volume, you automatically regulate your sodium levels. Okay, so here are some of the mechanisms that regulate sodium and water balance and maintain blood pressure homeostasis. So let's start out with a lowered systemic blood pressure or volume and take a look at some of these different things that happen. So we're going to start over on the right with ADH release and effects. So the posterior pituitary will release antidiuretic hormone, which will affect the collecting ducts of the kidneys, and so it will cause them to place aquaporins in the membranes of the collecting ducts of kidneys that result in greater H2O reabsorption. Next, we look at neural regulation, and so a decreased systemic blood pressure or blood volume will inhibit the baroreceptors that you find in blood vessels. They'll send signals, and the sympathetic nervous system will respond by causing the vasoconstriction of systemic arterioles. Now, this is going to result in an increased peripheral resistance. And now we're going to take a look at the renin-angiotensin-aldosterone mechanism, and that one's pretty complicated. And so decreased filtrate sodium chloride concentration in the filtrate that's in the ascending limb of the nephron loop, or a decreased stretch in afferent arterioles. Either one of those can cause the granular cells of the kidneys to release renin. And what renin will actually do is, down the line, cause the conversion of angiotensinogen to angiotensin 1, and ACE, or angiotensin converting enzyme in the lungs, will convert angiotensin 1 to angiotensin 2. Now, there are a couple of chains of events that occur as a result of angiotensin 2. In one, it will affect the systemic arterioles, causing vasoconstriction, resulting in greater peripheral resistance. Secondly, it will cause the adrenal cortex to secrete aldosterone that will target the distal convoluted tubules in the kidneys and cause greater sodium and therefore water reabsorption. So what you're going to see is increased peripheral resistance as one of the two sort of major responses to this, and also increased water reabsorption, and all of these together will, again, so you'll see peripheral resistance and sodium and H2O reabsorption, and again, all of these put together, sorry, let me get past the arrows here, all of these together will result in a greater blood volume and therefore an increased blood pressure. So what I want you to really look at is how all of these systems actually work together. Okay, the regulation of potassium balance is in the distal convoluted tubule, and we alluded to this in the last video, the distal convoluted tubule and the collecting duct. So it's controlled in the cortical collecting ducts. And so these are going to be the portions of the ducts that are in the cortex, and this regulates the amount of potassium that actually gets secreted into the filtrate. If you'll recall, I stated that potassium gets secreted into the filtrate via an antiport protein, and so how active is that antiport protein? It gets regulated, and it kind of determines how much of potassium gets secreted into the filtrate. So if you have a high potassium content in the extracellular fluid, then the principal cells will secrete potassium. Lower levels will cause the same principal cells to reduce the secretion of potassium to a minimum. In addition, if you're low on potassium, type A intercalated cells can also reabsorb some potassium that's left in the filtrate. So it's not just about reducing secretion. At a certain point, you can also reabsorb some of the potassium that's left in the filtrate. So the kidneys do have a limited ability to retain potassium. it means that most potassium is lost in the urine, and it may lead to deficiency if it's not replaced in the diet. What's the influence of aldosterone? It stimulates potassium secretion and thereby sodium reabsorption by principal cells. So the adrenal cortical cells are directly sensitive to the potassium content of extracellular fluid, and what this means is they don't require outside signals. They can directly detect potassium content. And so increased potassium in the adrenal cortex will cause the release of aldosterone, and this will increase potassium secretion. So again, it means that potassium controls its own extracellular fluid concentrations because there is feedback regulation of aldosterone release. So you don't have to go through the central nervous system to take care of this. All right, so salt substitutes have become popular. It's an effort to reduce sodium chloride levels that affect blood pressure, but the problem is salt substitutes are high in potassium, and heavy consumption of this, if you really like salt, you better not have too much of potassium chloride because it's only safe when the aldosterone release in the body is working normally. This means that abnormalities of aldosterone can cause problems with potassium levels. And I'll just tell you all this, KCl or potassium chloride, that is used as a salt substitute, it's also used as a part of lethal injection. So in the absence of aldosterone, hyperkalemia is swift and lethal regardless of your potassium intake. So aldosterone is really important in controlling your potassium levels. Now, if you have an adrenocortical tumor, and this puts out a lot of aldosterone, well, what's going to happen here is that extracellular fluid potassium levels get so low that neurons all over the body hyperpolarize, and this will cause paralysis. So as far as calcium and phosphate goes, sorry about the top of the slide, that should say regulation of calcium and phosphate. 99% of the body's calcium is found in the bones as calcium phosphate salts, and the balance is controlled by parathyroid hormone, which you saw earlier this semester in the endocrine system, and it doesn't deviate from normal limits very often. So parathyroid hormone will promote an increase in calcium levels by targeting bones, and the cells specifically that it targets are osteoclasts, which break down bone matrix and release calcium and phosphate to the blood. They'll cause the kidneys to increase their calcium reabsorption and decrease phosphate ion reabsorption. And in the small intestine, they will increase calcium absorption, but they do it indirectly because they stimulate the kidney to activate the vitamin D precursor that's present in your bloodstream. So chlorine is the major anion that accompanies sodium in extracellular fluid and thereby helps maintain the osmotic pressure of blood. 99% of chlorine is reabsorbed if you have a normal pH, because it passively follows sodium in the proximal convoluted tubule, and also because it's coupled to active transport of sodium in other tubule segments. So you'll recall from the last video that you saw a lot of sodium-potassium pumps. It means that where you've got active transport of sodium occurring, you've got passive transport of chlorine following it. If you have acidosis, you get fewer chloride ions, and you have more bicarbonate ions reabsorbed. Other anions do have transport maximums, and their excesses do get excreted in the urine. Examples of these are sulfates and nitrates. Now, pH affects all protein functions, all biochemical reactions, and therefore it gets closely regulated by the body. And so this is part of our acid-base balance. The normal pH of body fluids for arterial blood, the pH is about 7.4. For venous blood and interstitial fluid, the pH is at about 7.35, and we talked about why when we talked about the circulatory or the respiratory system, in that a higher proportion of CO2 and therefore carbonic acid results in a lower venous blood pH. So in intracellular fluid, the pH, normal pH is about 7. So you get alkalosis or alkalemia when your arterial pH exceeds 7.45. You get acidosis or acidemia when your arterial pH drops below 7.35. So you can see here that there's a fairly tight control over acid-base balance. Now, small amounts of acidic substances can enter the body in food, but most hydrogen is produced as a byproduct of metabolism. So if you'll recall way back from last September, or oh my gosh, I guess last September or October, when we were talking about cellular respiration, when glucose gets oxidized, it's going to give up both electrons and hydrogens. So you've got a major source of hydrogen ions just as a normal byproduct of metabolism. So a phosphorus-containing protein breakdown will also release phosphoric acid into the extracellular fluid. So this is another source of acidity. You also get lactic acid from anaerobic respiration of glucose. You get fatty acids and ketone bodies from fat metabolism. And again, hydrogen gets liberated when CO2 gets converted to bicarbonate in the bloodstream. So the concentration of hydrogen ions gets regulated by three mechanisms. First, by chemical buffer systems, and it's your first line of defense. Secondly, by brainstem respiratory center, so this is your second line of defense that acts within 1 to 3 minutes. And third, renal mechanisms, which are the most effective, but they do require between hours and days to cause pH changes, and you may not have that much time. Now, the difference between strong and weak acids is that strong acids will dissociate completely in water, meaning that they completely give up their hydrogens, and this can dramatically affect pH. Now, weak acids only dissociate partially. It means that they don't fall apart into hydrogen and an anion. So these are efficient at preventing pH changes. They can actually be used as a buffer. So strong bases will dissociate easily in water. They will very quickly tie up hydrogen ions, and weak bases will accept hydrogen ions more slowly. So if we take a look at this, a strong acid like HCl dissociates completely into its ions. So you can see in the solution there, nothing but hydrogen ions and nothing but chlorine ions. But if you take a look at carbonic acid that's over on the right, you'll see some of the carbonic acids holding together and not dissociating. That's the difference between a strong acid and a weak acid. And so what I want you to remember is that pH is basically a measure of the free hydrogen ion concentration. And as long as hydrogen is bound up in H2CO3, it's not free, and therefore it doesn't contribute to the pH. So a chemical buffer system is one or more compounds that help resist pH changes when you have a strong acid or base added. And so basically chemical buffers will bind to hydrogen if your pH drops, or they'll release hydrogen if pH rises. And you've got three major buffering systems. One is the bicarbonate buffer system. There's also the phosphate buffer system and a protein buffer system. So the bicarbonate buffer system is a mixture of H2CO3, carbonic acid, which is a weak salt, and also bicarbonate salts. So a bicarbonate salt example would be sodium bicarbonate, which is a weak base. So bicarbonates will buffer both intracellular fluid and extracellular fluid, but it's the only important extracellular fluid buffer. If you add a strong acid, then the bicarbonate will tie up hydrogen and form carbonic acid. And you see that happening in this reaction. So your strong acid, which is HCl, your weak base, NaHCO3, you end up with a weak acid and a salt. So this is something that might prove useful for you if you're prone to acid reflux or indigestion. So of course you've got hydrochloric acid in your stomach. If you stir some baking soda into water, then it will combine into carbonic acid and also into salt. So you're basically turning the gastric juice containing hydrochloric acid into, well, something a lot closer to salt water. The other thing that can happen there is that H2CO3 can also split apart into water and CO2. So when you do this, you're going to get some CO2 bubbles and that will come up as burps, which for me is kind of part of the fun. Now, the pH will only decrease slightly unless all of your available bicarbonate is used up, and this is something that's called the alkaline reserve. Basically, let me clarify this a little bit more. If you add a strong acid, then because of the bicarbonate that's in your bloodstream, your pH will only decrease slightly. Okay, so your bicarbonate levels get closely regulated by the kidneys. If you do add a strong base, it causes H2CO3 to dissociate. It'll donate hydrogen itself. So this is something that you can see happening. Your pH will only rise slightly, and the hydrogen ties up the base. So if you look at this, sodium hydroxide is a very strong base. To give you some perspective on that, it's an ingredient in oven cleaner. So if you add that to a weak acid, then what you're going to get is and I'm sorry, I don't know what happened to the text here, but you're basically going to get a salt of that weak acid that should be NaHCO3 and H2O. So I apologize for that faulty diagram. So your H2CO3 is almost limitless. It's because you have CO2 released by respiration. In fact, you get enough CO2 released by cellular respiration where you have to get rid of it, as much of it as you can. So you're not going to run out of carbonic acid. Okay, the phosphate buffer system has an action that's nearly identical to the bicarbonate buffer, and you have sodium salts of dihydrogen phosphate, which is a weak acid. So instead of being H2PO4, it would be Na2PO4. Also, you can have a sodium salt of monohydrogen phosphate, and so again, it's going to be NaPO4, which is a weak base. These are not really important in buffering plasma, but they are an effective buffer in urine and in intracellular fluid, where you have higher concentrations of phosphate than you do in extracellular fluid. So in the phosphate buffer system, hydrogen that's released by strong acids gets tied up with a weak acid. Okay, so it'll basically transform or be converted to a weak base. Okay, so strong bases also get converted to weak bases. So basically the thing that I want you to see here is HCl at the bottom, and I'm sorry, it looks like the order's screwed up on this slide again. So HCl at the bottom, which is a strong acid, it can be converted to a weak acid by a weak base. Okay, with the protein buffer system, you have intracellular proteins and also plasma proteins, which are amphoteric. Basically, they're big enough to where they've got groups that can function as both weak acids and weak bases. And what that means is that when the pH rises, these buffer proteins, their carboxyl groups can release hydrogen, and if the pH falls, then their amine groups can bind to hydrogen. Hemoglobin, among other plasma proteins, can function as an intracellular buffer. So you've got the bicarbonate buffer system, which is a mixture of a weak acid and salts of bicarbonates. It's your main extracellular fluid buffer, and it also operates in intracellular fluid. The phosphate buffer system is salts of phosphoric acid, which is a weak acid, and those salts are weak bases. And it is an important buffer in both urine and in intracellular fluid. And again, I don't know what's happening with the number 22 there. It shouldn't be there, so I apologize for that. In the protein buffer system, you have some amino acid side chains that can act as weak acids. Others can act as weak bases. This is your most important buffer in intracellular fluid, but it also plays a minimal role in blood plasma. Now you can have respiratory regulation of hydrogen. So alkalosis will depress the respiratory center, and the rate and the depth of breathing will decrease, and this will cause the hydrogen ion concentration to increase. So a respiratory system impairment can cause acid-base imbalances. And so that's actually a danger of the coronavirus that we're facing right now. So hypoventilation can cause respiratory acidosis, and hyperventilation can cause respiratory alkalosis. So breathe too little and you get acidosis. Breathe too much, you get alkalosis. So chemical buffers can't eliminate excess acids or bases from the body. The way that the lungs work is to eliminate the carbonic acid by eliminating CO2. And that way the only thing that's left over is H2O. The kidneys will eliminate non-volatile acids, and this can help prevent metabolic acidosis. The kidneys can also regulate the blood levels of alkaline substances, and they will renew chemical buffers. Now, in order to maintain your alkaline reserve, so these bicarbonate ions, the kidneys have to reabsorb bicarbonate. As it turns out, the tubule cells are impermeable to bicarbonate, but they are permeable to CO2. And basically this means that bicarbonate doesn't come back into the body directly, but it crosses through cell membranes as CO2 and then can be converted with the reaction from CO2 and H2O being converted to bicarbonate. So once it comes back into the cells, the CO2 can be converted back to bicarbonate, or it can leave the cell as CO2. So the mechanism here is coupled to hydrogen secretion, and basically the idea here is that for every hydrogen that gets secreted into the filtrate, bicarbonate gets reabsorbed. Now, reabsorption of filtered HCO3 bicarbonate is coupled to hydrogen secretion, and we're going to see how here. So I mentioned that CO2 crosses the cell membrane pretty easily, and we're going to start from there. And the CO2 that's within the cell combines with water inside the cell, and it's going to form H2CO3, which you see outlined in the green circle. Now this H2CO3 gets split into both hydrogen and the bicarbonate ion. So the hydrogen here gets secreted into the filtrate, and the HCO3 gets secreted, and basically in its secretion it enters the peritubular capillary. Now this is going to enter either, as you can see here, symport with sodium in the same direction as sodium, or antiport with chlorine in the opposite direction from chlorine. Now in our next step out in the filtrate lumen, so look over to the left, the secreted hydrogen will combine with bicarbonate in the filtrate, and what this will do is to form carbonic acid. So basically bicarbonate disappears from the filtrate at about the same rate that bicarbonate enters the peritubular capillary blood. So this carbonic acid that gets formed in the filtrate will then dissociate to release CO2 and H2O. And from here, the CO2 can diffuse right back into the tubule cell, where it starts another round of hydrogen secretion. So metabolizing food will generate new hydrogens. So as you break it down, you release hydrogens, and this has to be balanced by generating a new bicarbonate. And the way that you do this is that, again, secreted hydrogen gets used to reclaim filtered bicarbonate. So that means that the secreted hydrogen doesn't really get excreted from the body. It gets incorporated into an H2O molecule. So if you'll recall from that very last slide, you had hydrogen combining with bicarbonate to form carbonic acid, which split into CO2 and H2O. Now that H2O can be reabsorbed. And what it means in practical terms is that the body has the same number of bicarbonates and hydrogens that it started out with, and you don't have any net gains. So let's take a look here at how bicarbonate gets generated by the buffering of secreted hydrogen by monohydrogen phosphate. So first thing here, take a look at the inside of the cell. So inside the cell, CO2 combines with the water, and it will form carbonic acid. In our second step here, the carbonic acid gets split into hydrogen and to bicarbonate. And at the same time, the hydrogen gets secreted into the filtrate by a hydrogen ATPase pump. In the meantime, for every hydrogen that you secrete in HCO3 actually enters the peritubular capillary by way of an antiport carrier. Now the hydrogen that's secreted into the lumen will combine with monohydrogen phosphate, and this will form phosphoric acid, and this gets excreted in the urine. So the upshot here is what you're doing is you're generating a new bicarbonate. Another way of generating new bicarbonate ions is via NH4 excretion, and so this is a more important mechanism for excreting acid, but what it does involve is the metabolism of glutamine in proximal convoluted tubule cells. So each glutamine will produce a new NH4 and two new bicarbonates. So the bicarbonate moves to the bloodstream and the NH4 gets excreted into urine. So again, what this does is to replenish the bicarbonate levels, in other words, the alkaline reserve of the blood. So here, the proximal convoluted tubules, so see in step 1, they metabolize glutamine to NH4 and bicarbonate. That the NH4 gets secreted into the filtrate, and that takes the place of hydrogen in a sodium hydrogen antiport carrier. So it can basically kind of move through the same antiport carrier that hydrogen does. You see it moving here, sodium in, ammonia out. Now for NH4, each one that you secrete, a bicarbonate ion enters the peritubular capillary by that symport carrier, so you can see with those dashed arrows that it moves in the same direction as sodium. Now in the meantime, the NH4 gets excreted out in the urine. So all imbalances in acid-base balance get classified as either respiratory or metabolic, and the reason why is because you can have acidosis and alkalosis caused by both respiratory or metabolic causes. And so in respiratory acidosis and alkalosis, it's caused by the failure of the respiratory system to perform its pH balancing role. And your most important indicator that that's going on is your blood carbon dioxide levels. In metabolic acidosis and alkalosis, this is all abnormalities other than those that are caused by higher CO2 levels in the bloodstream. And so these can be indicated by abnormal bicarbonate levels in the bloodstream. So your most important indicator of respiratory function is the level, partial pressure of CO2, which is normally 35 to 45 millibars. Now, if you get above 45 millibars, this is going to cause respiratory acidosis, which is the common cause of acid-base imbalances. And this can be due to a decrease in ventilation, so just not breathing as much, or a decrease in gas exchange as in emphysema, pneumonia, or cystic fibrosis, and I guess now coronavirus. And what happens here is that CO2 accumulates in the bloodstream, and because that CO2 can combine with water to form carbonic acid, the blood pH will drop. Now, a PCO2 below 35 millibars is going to result in respiratory alkalosis, and this can happen as a result of hyperventilation, either due to stress or to pain. And what happens in hyperventilation is that you breathe out CO2 faster than you produce it. Now, metabolic acidosis is going to be indicated by a low blood pH and bicarbonate levels, and your causes can be the ingestion of too much alcohol, and the reason why that causes acidosis is because it converts to acetic acid. An excessive loss of bicarbonate can happen in persistent diarrhea, or the accumulation of lactic acid under exercise or shock, ketosis in diabetic crisis, starvation and kidney failure. So these can be causes of metabolic acidosis. Causes of metabolic alkalosis, which is indicated by rising blood pH and bicarbonate levels, First thing to say about this is that it's much less common than metabolic acidosis, but causes can be vomiting of acid contents of the stomach or an intake of an excess base, and an example of this could be an overingestion of antacids. A blood pH that drops below 6.8 can cause a depression of the central nervous system, leading to coma and to death. A blood pH above 7.8 can cause an overexcitation of the nervous system. This can lead to muscle tetany, nervousness, convulsions, and death from respiratory arrest. So neither one of these is a good alternative at all, and it's the reason why your blood pH is definitely a homeostatic variable. If the acid-base balance is due to the malfunction of one of the buffer systems, another system can try to compensate for that. So the respiratory system will attempt to correct metabolic acid-base imbalances, and the kidneys will attempt to correct respiratory acid-base balances. So in respiratory compensation, the lungs will try to compensate for metabolic pH problems by changing the rate and the depth of breathing. So in metabolic acidosis, again, the respiratory system will compensate by increasing the depth and rate of breathing. So that's the reason why high hydrogen levels can stimulate the respiratory centers. And a metabolic acidosis with respiratory compensation gets indicated when the blood levels below 7.35 pH, and you've got lower levels of bicarbonate and PCO2 levels. And the reason why the PCO2 levels are below normal is because the lungs are going to be blowing off more CO2. And if more CO2 is being exhaled, then that means there's going to be less combination with water to form carbonic acid, therefore less bicarbonate present. So in metabolic alkalosis, the respiratory system tries to compensate by decreasing breathing, which will allow the CO2 to accumulate in the blood, and that will lower the pH. And so your indicators for metabolic alkalosis with respiratory compensation is a high pH due to elevated bicarbonate levels and a PCO2 above 45 as your lungs try to build these hydrogen levels up. Now, renal compensation can try to compensate for pH problems caused by the lungs, and they will adjust bicarbonate levels. So in respiratory acidosis, the kidneys will reabsorb more HCO3. And so this causes you to secrete more hydrogen. And so the way that this works is that hypoventilation or low breathing rates can cause an elevated CO2 level. And so again, your respiratory acidosis with renal compensation, what are your indicators that your acidosis is caused by the respiratory system? A low pH of the bloodstream, a high PCO2, and high bicarbonate levels. Now, when you've got respiratory alkalosis, the kidneys will secrete more bicarbonate. And so the sign that your alkalosis is respiratory rather than metabolic is high pH, a low PCO2, and decreasing bicarbonate levels. So one of the problems that you've got here is that the respiratory system can't compensate for respiratory acidosis or alkalosis. It's the root of the problem and can't serve as a compensation for it. The renal system cannot compensate for metabolic acid-base imbalances, and so these two systems have to compensate for one another.